Colorectal cancer is a common and deadly carcinoma of the gastrointestinal tract that occurs due to an accumulation of different mutations. It is important to remember that this cancer, like all other cancers, takes a long time to develop. Colorectal cancer accounts for 12% of all new cancer cases in Canada. In 2020, it is estimated that each day, 73 Canadians will be diagnosed and 27 will die from colorectal cancer. There are modifiable and non-modifiable risk factors associated with colorectal cancer. Modifiable risks include smoking, an increased intake of red meats, a lack of fiber, and obesity. Non-modifiable risks include being elderly, male, having IBS, familial adenomatous polyposis, and hereditary non-polyposis. The GI tract lining is made of 4 layers: the mucosa, submucosa, muscularis, and serosa. The innermost layer, the mucosa, is made up of epithelial cells. These cells are constantly being destroyed and regrowing every 6 days. The rapid growth of cells occurs when a variety of genes malfunction. One of the first mutations occurs in a tumor-suppressing gene called APC. Normally, the APC gene stops the cells from growing and dividing too fast, but when there is a mutation in the gene, the epithelial cells can divide and grow without any restriction, creating a polyp. The RAS oncogene is a G protein that controls cell division by transmitting signals to indicate when the cells should divide. When this oncogene is mutated, it allows the cells to continue dividing even when there are no proteins to activate the system. It is also believed that there is a loss of a tumor-suppressing gene called DCC. DCC is a protein receptor that binds onto a protein called netrin-1. When they are bound together, this promotes cell survival, but eventually the netrin-1 should be released from the DCC receptor to allow for apoptosis. In colorectal cancer, a mutation occurs causing the loss of this gene, which allows the cells to continue dividing. This rapid growth results in the formation of a larger, normally benign structure called an adenoma. As time goes by, more tumor suppressor genes are lost, including the gene that codes for the p53 protein. The mutations in all of these genes contribute to the formation of growths in the colon and rectum. Like other cancers, colorectal cancer develops in stages. Stage 0 is when the growth is small and doesn't extend past the mucosa. Stage 1 is when the growth is beyond the mucosa but not into any lymph nodes or other organs. Stage 2 is when the tumor infiltrates the whole rectal wall and it may reach nearby organs but not lymph nodes or distant organs. Stage 3 is when the tumor has spread to lymph nodes but not distant organs. Stage 4 is called metastatic, so the cancer has reached distant organs. Symptoms depend on where the growth is in the colon or rectum. When a growth is in the ascending colon, it is beyond the mucosa and pain and weight loss occur. Depending on the stage, a palpable mass can be felt, but many remain asymptomatic until the growth is very large. When a growth is in the descending colon, the mass typically causes lumen narrowing, which leads to early bowel obstruction. Growths in the descending colon are more common than growths in the ascending colon. Wherever the mass is, pain and bleeding can occur. Blood will be found in the stool, which is known as hematochezia. Homeostasis is disrupted due to the rapid proliferation of the mucosal cells in the GI tract. The DCC protein receptor, normally found in the cell membrane, is lost, which results in the formation of an adenoma. In colorectal cancer, the DCC, P53, and RAS proteins are not functioning properly, which contributes to the rapid growth of cells.